All right, thank you for the introduction. I was going to kind of go through this sequentially, but I think with this group, I think I could go through it all. We could take, have a conversation at the end about sequencing technologies, but I also think that it might be nice to just, as we go, feel free to just ask questions and we can just sort of discuss things as we go. So I'll just get right into it. In my time here, certainly, which has been, seems like forever, we've seen like this incredible sort of scale of human genome sequencing or just sequencing in general. The Broad Clinical Labs is actually, I should specify this, is sort of the rebranded name for the Genomics platform. That's been a big part of Broad data generation for a really long time. As we've stepped closer towards sort of like clinical offerings, we really just needed to like have, the world aware that we don't just do sort of like, the human genome project style projects anymore. We really are stepping towards sort of clinical genomics, and it's been an exciting time. But as you can see, if any of you have visited the 320 Charles Street site, it truly is a genomics factory. We've been generating, tons of data for many years, and these are some exciting plots, and these are presented all over the place. But it really does show the sort of the exponential increase in sort of sequencing output that's happened here at Broad. If you just look at, petabytes of data generated, we're talking about, almost 100, and that's really just been in the past, 10, 12 years. The number of genomes that we've sequenced, well over 500,000 whole genomes deeply sequenced, almost 1 million exomes, and tons of arrays and RNA-seq. And something that we're particularly like sort of proud of, and I think it's sort of like a testament to the group and what they're able to do is, almost like a bunch of those genomes that we've sequenced, a lot of that 500,000 actually were done between 2021 and 2022 while we were, the team was simultaneously scaling COVID diagnostic testing. So we ended up sort of pivoting everything that we were doing to COVID diagnostics, but as some of these big projects came online, we were able to continuously, sequence human genomes. And we've contributed, to a lot of great projects over the years. And these are more just some of the recent ones. The All of Us program is a large federally funded program to sequence 1 million Americans, bring their genomes to 30x coverage, do array work, and then do subsequent studies after that with that data set. We've done, several 100,000 of those genomes. We'll be slated to do upwards of 600,000 for the total program. We've supported some of the large population neuropsychiatric disease studies, obviously supported, the CDC and their efforts for COVID sequencing. And then, a lot of other things like microbial work, RNA-seq, targeted panels and such. So, you've probably seen this plot before. I like it because it almost spans my entire career here. So I feel like I can speak a little bit about this curve. Sequencing costs don't just drop randomly. For the sort of initial human genome project efforts, a lot of the sequencing is done with, Sanger sequencing. So I don't know if people are familiar with, people still use it quite a bit, but not in generating the large population scale studies. They use it to, confirm a lot of variants that are found in clinical studies and things like that. But Applied Biosystems was the group that always sort of led the charge in sort of advancing technology up until about 2006, 2007. And you just got this flurry of activity around what now everyone's calling next generation sequencing. It's back in 2007. I feel like next generation sequencing, that term is just sort of past at this point. It's just kind of just sequencing as we do it. But a lot of different groups popped into the market and we're really presenting alternatives to sort of, capillary electrophoresis separation of fragments to discern, base resolution. At that time, Solexa, which is now Illumina, emerged. Ion Torrent, another group called 454, Helicos, and there was a bunch of other ones that kind of came into the market and kind of disappeared. And a lot of these kind of still exist and really are useful for certain applications in different types of labs with different types of utility. But that is what drove, this inflection point in 2007 to really start making the cost of a human genome, to start descending. and that lasted for a good number of years. But really, there was like one winner that came out of this, and it was Illumina. And I think most of the projects that you guys are involved in, if you're generating lots of sequencing data, a lot of that data has been generated probably with Illumina sequencing. And I'm going to talk a little bit about Illumina sequencing because it's still the workhorse of a lot of what we do. But as you can see, with one big player in the market, the costs really have plateaued a little bit. And forever we've been talking about, the promise of the $100 genome. And we've been kicking around this $1,000 range for quite a while now. But now in like 2022, 2023, over the past couple of years, I mean, a lot of new players are in the market. And so, the Genomics platform, or Broad Clinical Labs now, is really invested in understanding a lot of these new technologies that really are, one, pushing down the cost of data generation, but also enabling different types of analyses to happen. And I'll get into that as we sort of, move through here. Sorry. So, today we have a very large fleet of instruments. And yeah, I've sort of alluded to the fact that we're still doing tons of data generation with Illumina, but we have their full sort of suite of instruments, from very, very small benchtop systems to like the mega systems, which is the NovaSeq X. But we're also, now onboarding long read PacBio instruments, where we've been messing around and, optimizing workflows for the Oxford Nanopore technology. And also there's another one that I'll talk about, which is this Ultima Genomics, which is short read technology, and also Element as well. The Genomics platform has always been in this like sort of exciting position to be able to, onboard and look at new technologies as they emerge, or even before they really emerge onto the market. A lot of the technology groups want Broad to sort of assess their technology and work with us to sort of understand how it would be best be applied. So we always get these sort of early looks, which is fun. And we have this big suite of different types of sequencing technologies because we're really trying to support not just one large project like the All of Us study, but we do cancer genomics, we do expression analyses, we do population, GWAS studies, and the whole bit. So we have this, very large sort of menu of offerings that we're we need to support by different sort of technologies that fit those different sweet spots of the different efforts. So, I mean, whole genome sequencing is a huge part of it, but we're also, clinical trial services and whole transcriptome analysis, and we're moving more and more towards like, how do we do large perturb-seq studies? The cancer field, where you're trying to identify very low allele fraction variants, you're really needing to sequence super, super deep, potentially in a targeted panel. And, cheap sequencing is a huge part of enabling that to happen. So, for the, maybe the first like 10 years of that slide I just showed on the cost decrease, a lot of that has been targeted towards research stuff, but we really are moving towards supporting research, preclinical studies, actual clinical research, and then also clinical diagnostics as well. And I think over the next few years, you'll hear more and more around the efforts to sort of incorporate a clinical whole genome into sort of medical records, and understanding how impactful that is in treatment and care. So, we can get just right into sort of the sort of like new sort of menu of technologies that we have. It would be silly not to start with short read technologies. And when we say short read, we're talking about, short fragments of DNA in the 500 to 600, base size, and usually reading in 100 or 150 bases from either side. So you really are just looking at these unique short little sequences that are then aligned to a reference genome. So Illumina, been around for a long time. It uses a method called sequencing by synthesis. The vast majority of data in probably all the databases in the world right now has probably been generated by Illumina sequencing. And they really do, provide platforms that, that span from like small benchtop systems that a small academic lab might use to, ultra high throughput, production scale systems that are, for example, today supporting the All of Us program. It remains a major player in the, the next generation space. And typically the way we're operating it in the most efficient way is running paired end 2 by 150 base reads, which has been sort of like the typical approach for whole genome sequencing, meaning you're reading in 150 bases from one side of the fragment, and then you reverse and you come in the other way and read that, and then those two reads can be aligned to the genome. And so the way it actually works is you have this very customized consumable, which is called a flow cell. And inside that flow cell, you have individually addressed lanes that you can take a sample that's been fragmented, and you've appended these sort of, or ligated on these adapters to the end that make that fragment of DNA capable of binding to this sort of lawn of oligos that are plated, millions of them across that sort of glass channel there. And once a fragment is actually bound to the glass, a polymerase can come in and start, priming off the oligo on the glass and start adding base by base. And in this case, it's pretty straightforward. The bases, the nucleotides that are added have a fluorophore on them. A, C, G, and T have different fluorophores. You incorporate one at a time, so they're blocked. So you flow in a mixture of all the nucleotides, and anywhere it finds its complement, it's added. You read the entire flow cell, you get the color that's interpreted into sort of a base call. And then what happens then is the blocker is reversed so that now another nucleotide can be added. So the fluorophore is cleaved off, another nucleotide is added. It just goes step by step by step, and eventually you have a 150 base sequence that's been called into actual, bases of sequence, and that's just, computationally aligned to the genome. Straightforward. It's been a great technology for a really long time. If we look at like the existing state of today, there's an entire sort of quiver of Illumina sequencers people can choose from. We usually end up onboarding and assessing all of them that they release, but still to this day, the NovaSeq X, which was released really in the past probably like year, we're onboarding it right now, and this truly is the workhorse of sequence generation. I mean, this one run of the NovaSeq X can generate up to 64 whole human genomes per run in about 48 hours. If you think back to the original human genome, which took years and years and years and $1 billion, it really is amazing, like how quickly a genome can come off these machines. And we're running these now. We're onboarding them. We're running them pretty much continuously to support both research and clinical genomes. We still run a lot of exomes, and that this new product, which is is called blended genome exome, which is a genome backbone, which is about a 4x PCR-free genome, usually around a 3x actually, with a higher coverage of all the coding regions of the genome. That's becoming a very popular product, and people are excited to run that. But again, to run these as cheap as possible to enable more science to happen, it's about driving down that per gigabase cost. And the NovaSeq X really has driven down that cost quite a bit. Of course, cancer genomes and targeted panels in cancer, you really do want to sequence deeply to be able to tease out the variants that are present either in a tumor at a low fraction or even in liquid biopsy. And of course, we're doing lots of RNA-seq and transcriptomics as well. Any questions about Illumina sequencing? I feel like a lot of you who will engage with the platform will probably generate Illumina data, but it's not your only option. So we can get into some of the new ones. So over the past year, a few more of these companies have emerged. Element Biosciences, which is it's actually a really great group that a lot of folks who had been at Illumina started this new company with sort of an optimized approach to how you read bases as you're sort of adding them one by one during sequencing. So the general concept is pretty similar. It's still 2 by 150 base reads. You can get about maybe 300 gigabases per flow cell, which translates to about 2 genomes. And there's 2 flow cells that can run on this instrument simultaneously. So that gives you about 4 whole genomes per run and about a 38-hour runtime. And it really, what this new technology has allowed is it actually enables a pretty competitive price per gigabase of sequencing for sort of a mid-throughput system. So for example, to leverage, sort of the really cheap data generation from the NovaSeq X, you need a lot of samples to run to get that sort of super cheap rate. With Element, however, they really are offering a system that can allow you to do a smaller study with pretty comparable, costs, in data generation. So you don't need a 10,000, person study to actually justify the cost and be able to multiplex that many samples. Processes like microbial sequencing, where you might only, like for microbial isolate, you might only be interested in generating a half a gig of data. In this situation, you're probably not going to load that on a HiSeq X. This system provides, pretty cheap sequencing for some of these smaller, more niche groups. And we're offering this today through our walk-up sequencing workflow. And so,
Brendan, sorry to interrupt. There had been a question from the audience about, with these kind of high throughput sequencers, do you ever worry about sample cross-contamination with the multiplexing? For example, if you have cancer genomes on the same run as a prenatal cell-free DNA sample, where maybe, you have a limited number of reads in the latter, I guess, do you consider sample type when you're multiplexing?
Yeah, definitely. And typically we will multiplex like samples together. One way to, that all these technologies have addressed, I think this is a great question, have addressed this sort of worry about, contamination. It may not be contamination of the sample way upstream. There's always worry that sort of like on the slide or, in the sequencer during amplification, you can get like a rogue fragment that gets amplified and attributed to say another sample that's multiplexed. What we do is we use an approach, and most of these groups are doing it. It's called dual unique indexing. So every fragment of DNA that we actually load onto these machines has a unique barcode at the beginning of the fragment and a unique barcode at the end of the fragment. And that's informatically registered before you start the work, so that reads have to have both of those barcodes that have been predetermined to actually be binned and associated with that sample that What this does is it keeps a partial fragment from priming off something else and incorporating like a different barcode. If you see a mismatch of barcodes, you just throw that away. It becomes an orphan read, and it doesn't get included. So only perfectly matched read pairs will actually be associated with a sample, and that really gets away from any sort of worry about contamination. Obviously, up front in lab workflows, you have to be extremely careful about contamination on the actual physical level. Moving liquids around, or if you have the tumor next to the normal, you wouldn't want those sort of like cross-contaminating. That's for sure. Does that answer your question?
That was great. Thank you so much.
Okay. So Element Biosciences, we're pretty excited about this technology. There's a lot of advancements here. They're referring to it as, if you go on their website, they refer to it as avidity, sequencing by avidity, which I do think is a completely made up word. And it's using a reagent that they're calling an avidite. So anyway, long story short, it's sort of like sequencing by synthesis, but instead of adding a nucleotide that has the fluorophore attached to it and then having to cleave that fluorophore and then add another nucleotide, what this does is it adds a nucleotide. Then separately, this quote avidite, which is sort of a multivalent complex of fluorophores and then recognition nucleotides, will come, flow past the fragment that's or it's actually like a clonal fragment. So it's been amplified on the slide. And you'll have multiple recognition sites to the bases that just been incorporated. You get a really bright signal. You're able to wash that away and then add another base without, quote, scarring the template. And so a challenge with traditional sequencing by synthesis is that every time you cleave that fluorophore from that nucleotide that's been incorporated and try to add another one after it, you kind of lose fidelity over time, or you can lose fidelity over time. And this just becomes a much cheaper reagent to use with a much higher signal to noise ratio on the platform. So we're getting very, very good base quality scores from this technology, and people are really excited about it. Any questions about sort of Element Biosciences? We right now we have 3 of these systems in-house. We're offering them through sort of like the walk-up path. So if people have sort of smaller smaller studies that you're not going to fill up something like a NovaSeq X. It would work well for that. Yeah.
So how does it handle poly A or poly T, like a simple repeats of the sequences compared to the Illumina sequencing?
I think it's a little bit better, and you know, short homopolymers, I mean, as they get super, super long, I think every technology is going to have a challenge with it. But in relation to sort of like that template scarring that can happen as you're cleaving off the fluorophores, since this is sort of more of a sort of like a native base that gets added, and then that avidite it integrates with it and then dissociates with it, the next base adding is a little bit more probably accurate, you know, as you move through a homopolymer.
So related to that, what about then high GC content sequence compared to the Illumina? Because the Illumina is known for, you know, having some troubles with very high,
course, like the extremely high
GC contents.
You know, I'd have to go look at that. I mean, we have a bunch of data, but I haven't looked at those plots. My guess is that for that same reason, it could be beneficial, but I feel like we need to look at the actual GC distributions to be able to answer your question like exactly. But it may be there. I just don't have it in this slide, but we should look at it. I think they suggest that it's improved. And there's another technology that I'll talk about in a minute that like is very much improved. And I think it's because of that sort of like less scarring sort of behavior. So Pacific Biosciences, and I'll be able to talk about PacBio a little bit more in the way most people are thinking about it, which is a long read sequencing technology. But they also acquired another group called Omniome, which is a short read technology that is very exciting because the base qualities are extremely high. So traditionally in DNA sequencing, whenever you get signal for a base, you're trying to call it ACG, you know, ACG or T. There's always some error rate that happens, and every base that's called is assigned a quality score, which we call a Q score. So a Q20 would say, we believe that there's probably an error 1 in 100 bases, Q30, 1 in 1,000, Q40, 1 in 10,000. Now, we haven't really seen like Illumina sequencing is generating many Q50 bases or Q40 and above, but this method, which is using a method called sequencing by binding rather than sequencing by synthesis, has a little bit of similarity, I think, to the element workflow. But what it does is it really is enabling super high, you know, base qualities. And in terms of the homopolymers, we do know that in homopolymers, it's very, very accurate. So the way that works is it's a little bit similar. It's not using an immediately like it's not incorporating the base that has the fluorophore right away. What it does is incorporates a blocked base, adds the base with a fluorophore that's complementary to the next base on the strand. That is dissociated after imaging, and then it's backfilled with that base that's blocked, and it just kind of goes in a stepwise fashion. So it's binding, removing, and then binding again, and it's continuing along. And again, what it leaves is an unscarred template that allows for very high accuracy. And again, we generated some whole genome sequencing data using this ONSO short read technology. And if you look on the left here, these are actually just, you know, plots across the 150, you know, base pair cycle. And in the colored lines are the actual read 1 and read 2 quality scores. And if you look at the ONSO, I mean, every single base is above Q40, extremely accurate. And if you compare that to Illumina, it's dancing around that Q40, and by the end of the 150 base read, it starts to sort of drop off a little bit down, you know, down towards like maybe high Q30s or something like that. And honestly, Q30 is great. For population studies, fantastic. But if you think about sort of the needle in the haystack applications, like liquid biopsy, looking for, you know, minimum residual disease or early detection of cancer from a blood biopsy and cell-free DNA, you're looking for the 1 in 1,000 event. Having a low sequencing error rate is extremely important for things like that. So would you necessarily sequence 100,000 whole genomes this way for a population study? Probably not. But for those applications, it's very, very exciting. And there's been a bunch of work that has been going on, working with the blood biopsy team to take like the method codec that's come out of Victor's lab, and understanding if you sequence it with a very low error sequencing technology, like what does that actually mean for early detection or minimum residual disease. And we've actually been able to, we've taken these standard control samples that are well characterized, this HG002, this sort of like telomere to telomere reference genome that they're building. And we sequenced that deeply using ONSO, and we're able to sort of resubmit about 1,300 sites for refining in that reference. So it definitely found a good number of errors that could be corrected to, really create a telomere to telomere reference genome of much higher accuracy. And that was the work Mark Flaherty and Laura had worked on for quite a bit. Now, I don't know if people have heard about sort of this new sequencing technology that just recently emerged called Ultima Genomics. This is actually where the technology I've been talking about have been sort of, I think, natural progressions of an existing technology over time. Ultima Genomics, that we've been sort of working as an early access partner with since, about 2019, is a very, very different approach. And their goal is to really just push down the per gigabase cost of sequencing with a different approach. So it's a flow-based sequencing. It's not incorporating a single nucleotide at a time that's blocked. It's actually flowing each individual nucleotide, A, C, G, or T, once at a time. And so if there is a homopolymer, you have 5 A's in a row, 5 A's are added, and then you're actually assessing that increase in signal from that event to discern whether there's, certainly more than one. But with all flow-based sequencing technologies, historically, it's struggled with things like homopolymers, because once you get out to 4, 5, 6, 7, 8, or beyond, differentiating between the signal between 8 bases added and 9 bases added truly is tricky. But if you take that aside, they're improving that, like with new models. But the neat thing about this technology is these machines are giant. They're like the size of like an enormous refrigerator. But what it does is it uses this modified silica wafer. All these other technologies I've been talking about use very, very specialized custom consumables, which we usually call a flow cell. They usually, have a top slide, a bottom slide. They're coated with all kinds of like oligos and things like that. You have to flow samples through them. This technology is simply using, commodity level silica wafers that are used for, every industry in the world. They're etched, so there's hydrophobic and hydrophilic regions, and beads are flowed across them. It's a completely open system. It's not enclosed. And the way you add one sample to the system or reagent is by spinning this thing like a record, like an old vinyl record, and dispensing to the middle, and everything just radiates sort of out across it. And what it's doing is it's getting you away from an extremely expensive consumable and using something that's just broadly available on the market, which is a silica wafer. When we started working with this, a lot of things end up with sort of code names before a company actually emerges onto the market. And Niall Lennon dubbed this, and this is the code name we kind of use internally, which was jukebox, because it really is like a stack of records that you're just kind of like sending through, dispensing a sample, and running across. So really, the Ultima Genomics really is striving to drive whole genome sequencing like even further down in costs, and it has been very exciting. Here you can actually see like that's the wafer. It's very simple, it's big, but it also generates a lot of reads. The way that it works is, I don't know that this technology is perfect for a small lab, but these are definitely for sort of industrial scale data generation. So it has these steps. I don't know if people have worked with emulsions before. So, aqueous solution emulsified with oil. So you get these like micro droplets within oil. And the idea is that you have these beads, these microscopic beads with a fragment of your genome attached to it. And you amplify those hopefully there's one bead and one fragment in each tiny micro droplet in this emulsion. And you eventually clonally amplify that. So there's plenty of copies to interrogate with nucleotides, but then you have to break that emulsion. There's very custom automation. There's custom robotics that do all this work. You know, there's a lot of oil. It's kind of I wouldn't call it a clumsy workflow. It's just an involved workflow, but it's kind of the stuff you have to go through to get that sort of bottom of the barrel cost per genome. But what happens is those beads, those emulsions are broken. You end up with a tube of just beads with clonal amplified fragments on them. That's injected onto the spinning wafer, and those beads sort of radiate outwards, just using centrifugal sort of forces, and settle in these sort of micro etched wells. And now you just have this big wafer with, you know, I should check exactly how many end up on it, but it's probably billions of beads or across the wafer. And then to add reagent to that, it's just shot on top of that thing. It spins. It incorporates a nucleotide and it images. Then you add the next base, same thing. You wash, and so you're just going through these constant cycles of adding stuff to this top, spinning it. It all shoots off to the sides, and then you image the whole thing. And it really is like today we have been to do work with Ultima. It's usually been sort of like a three-way collaboration between a researcher at Broad, Ultima, engineers and folks there, and then the genomics platform. So we've really been interested in looking at, we've done some sort of population-based whole genome sequencing. You know, SNPs and single nucleotide variants are called very, very well. Indels have a little bit of a challenge and some of the, because of the flow-based stuff, but that's improving, you know, really every day. I think a good application for this, it is single end reads. So you're not actually reading in from both ends of a fragment. So you can read about 300 bases in one direction, which does make sort of like alignment a little bit harder than paired end reading. One really interesting application that people are exploring here is very, very short read, sort of perturb-seq, where you're actually just doing digital counting. So counting applications where you might have sort of, you know, millions of reads, lots of single cells, with like perturb-seq, where you only really need to read maybe 50 bases to sort of assign a bead to a cell and sort of to an expression level. And so very, very fast data generation for something like perturb-seq is very, very attractive using this technology. Anyone have questions about Ultima? No? So again, this is sort of like a summary of the workflow, captures a little bit about it. You know, your normal sort of fragmented genome is you have adapters that are added to the ends of it, and then you go through this process of creating this emulsion, which is really just like making salad dressing at that point. You're adding your sample, you're adding oil, you vortex it like crazy, you end up with tons of these micro little droplets, hoping that you have one bead per, but obviously you get a distribution, some have two, but those are caught downstream. You PCR amplify that in an emulsion, you break the emulsion, and then, as I mentioned, you're dispensing it onto this open wafer flow cell. And here's just an example of like what single beads look like when they're actually, you know, with emitting signal with an interrogated base. Reagents are dispensed sort of through this sort of, you know, direct injection on top of it. And you have this scanning immersion imaging. So it's just sort of similar to how it's dispensed. You have this camera as your wafer, as your record is spinning, it's starting on the outside and moving sort of slowly in and in and in, imaging as it's going, which is a very new type of sort of detection. Those signals are sort of using optimized machine learning algorithms are trying to take that signal that's detected through imaging and convert it into actual like base calls, which are you know, computationally assembled into reads and then aligned against a reference genome. And then obviously variant calling happens after that. So that's sort of a summary of all the short read technologies that we're operating over in Broad Clinical Labs. Long read technology is becoming more and more important to understand. Long reads really do enable like a more comprehensive understanding of structural variation in the genome that a tiny little 500 base fragment can't really inform on. I think that as costs of long read sequencing come down, people are becoming more and more interested in doing more population scale stuff, because especially when you're looking at like sort of clinically relevant variants, I think there's more and more publications coming out there that are associating structural variants with disease that are really important to detect. And short read technology has always been good at detecting sort of SNPs and short indels and things like that, but has always struggled with long structural variants, duplications and insertions, deletions, and the such. And if you sort of look at this this one paper, looking at, you know, a pile of structural variants, long read sequencing was able to detect 25,000 or 2.5 times more than the short reads. And as the costs are coming down, we are like very much considering what population scale long read sequencing actually looks like. PacBio, which I talked about a little bit in the context of their short read new technology, PacBio has been playing in this long read game for a very, very long time. And while it's been relatively expensive over the past number of years, the costs are coming down very, very quickly right now. And this smart cell technology, the single molecule real-time sequencing, has been being optimized. It's been really well optimized over the past number of years, especially using this, what they call HiFi sequencing, which is single molecule circular consensus sequencing. So I'll try to describe it real quickly. If you look at, you start with high molecular weight template, so you can't start with super fragmented DNA to begin with. You need good quality, high molecular weight extracted DNA. You're shearing it, you're chopping it up, but you really want to chop it up into things that are larger than like 10 kb to efficiently use the actual sequencing that you're going to buy. Hairpin adapters are added to the ends of these, and you essentially create sort of like a circle, like a long loop. And during the last step of this process, after you add these sort of hairpin adapters, you anneal a primer and a polymerase to a recognition site in one of the adapters. And what happens is the way this technology works is you have this glass surface with an etched metal layer above it. And the micro well is referred to as a zero mode waveguide. And what the zero mode waveguide is, or the way it's described, is it's essentially a well that is so narrow, and the height is short and narrow enough that light can't, using the wavelength of sort of excitation that they use with a laser blow, can't make it all the way through this little pore, this little hole. And what happens is your molecule comes over, the polymerase sort of anchors to the bottom of the substrate, and that polymerase is acting as a motor, and it's incorporating nucleotides at a very, very, very high rate. As that sort of molecule is looped multiple times. So you don't just get a single look at a base. If you loop that 10 kb molecule 15 times around, you're actually, every base you're seeing, you're going to see 15 times. And if there's an error that happens, a misincorporation in one of those loops, you're able to actually develop a consensus. Say, well, like 14 times I saw an A, a single time I saw a G, we're going to call that base an A. And it really does allow for, when it comes to GC and homopolymers and things like that, it really is a good method to be able to create high quality long reads. And so this is just an example of, this is just looping around and looping around, and you end up with these long reads that can sort of be piled on top of each other. And then you end up with a sort of a HiFi consensus read at the bottom that has that sort of Q30 level of quality. The consumable is called a smart cell. It's down there on the lower right. The progress that PacBio has made in the past, even like over the past 18 months, we had upwards of 20 of the Sequel 2s, which was their most recent sequencer up until maybe a number of months ago, where they released the Revio, which really does it really does push the data generation up and the runtime down. So at the end of the day, it's about a 15-fold increase in output per instrument. Right now, a big driving project right now is we've taken about 10,000 of those all of us samples, and we're going to generate whole genome sequencing to about 12x for 10,000 samples. This also can support a lot of the work that's happening around isoform sequencing and things like that as well. One more before we kind of wrap up, I got about 5 minutes, is Oxford Nanopore. They're a cool company because they've made everything from a little sequencer that you can sort of keep in your pocket and walk around with or deploy in the field to very large systems like this PromethION, which can handle 48 flow cells at a time and actually is sort of now positioned to be able to do sort of larger population-based stuff. It's truly sort of long read single molecule sequencing in that your molecule is tagged with a motor protein, and that motor protein is attached to your DNA, brings it over to this pore, and it's actually driving, it's sort of splitting the double strand and sending one strand down through the membrane, down through this pore, and you're just measuring the disruption of this ionic current measured over time, and then, making base calls. Sorry about the typo, but it's running through at like 400 bases per second. And you can just keep running it and running it and running it. For whole genome sequencing, like we are interested possibly in sort of a clinical setting, but one thing that we're actually particularly interested in right now is this adaptive sampling that groups have developed, which is if you have a region of interest, so say you have a structural variant that you detected in a clinical genome that you want to confirm with an orthogonal method, you can say, I want to look particularly at these variants that I believe are in this genome or these regions of the genome. And what can happen is these pores can start reading a fragment and say, wait, in the first sort of, few seconds or something, first few hundred bases, be able to say, wait, does this align to the region I'm interested in? No. Okay, reverse that pore, eject that fragment out, and let's pull in another one and see if that one actually aligns with what we're interested in. So this allows this sort of like on sequencer enrichment for regions of interest. And right now it seems to be working pretty well because it is giving sort of a 4 to 6x enrichment of targeted regions over background. So we are exploring it for this sort of confirmation of SNVs from our clinical genomes that we sequence. Now, very quickly before I wrap up, so how do we know that these platforms work? All these companies advertise great performance and the whole bit. And what we've finally done is we've standardized, and this is a lot of work with Michael Gatson, we've standardized how we evaluate technology. So what we will immediately take sort of these control samples that are very, very well characterized in the world, we'll sequence them to depth, and then we'll take multiple technologies or even multiple conditions of the workflow that we might change in the lab, and we'll sequence these to depth, 30x genome sequencing, and we'll run it through this evaluation framework where we're comparing it to the truth and comparing it to sort of like that are their genomes in the bottle truth, and able to sort of truly come up with sensitivity and specificity for, say, SNPs and indels, but also divide those calls between sort of high confidence regions, low confidence regions of the genomes, high mappability, low mappability. So we have this now standardized workflow, and there's a whittle for it to be able to take new methods, bring them in, and understand their performance. And so, when we look at a bunch of these technologies that I've been discussing today, Illumina, Element, PacBio HiFi, which is long read, and ONT. I guess I don't have Ultima here, but you can see that we can run this analysis and say, yeah, these technologies are living up to sort of like the claims that the groups are making in their performance. All of them, if you look at it for SNPs and indels, for sure, all have very, very good precision and recall. This is just showing the F measure here. ONT over some of the low complexity regions obviously struggles a little bit. And that's just because of sort of highly repetitive regions are hard to discern, I guess, with like a flow-based sort of like workflow like that. So, and so a lot of folks here probably already engaged with Broad Clinical Labs for data generation, but if you're new or just joining the Broad, we have a walk-up sequencing model where you can create sort of libraries or experiments on your own and submit the sequencing ready sample to us, and we will just run it and return data back to you. Of course, you can work with a project manager, but you can also just email Genomics at Broad, or if any of your colleagues need sequencing services, we're available. And obviously I'm presenting this stuff today, but I mean, all that stuff that I've shown you is just, is a product of a gigantic team of really good folks over at the Broad Clinical Labs. And with that, I'll take whatever questions y'all have.
I'll start off the
Thank you.
questions. So it's amazing to see all this technology change over the past 20 years. Has there been the same type of fast-paced advancements in the library prep generation, like working with low input samples, things like that?
I would say that, early on when the cost of a genome was high, it wasn't such a huge concern. But as the cost of genome sequencing comes down, the relative cost of sample preparation actually starts to become a significant thing when you're talking about hundreds of thousands of samples. So there has been progress, but I wouldn't say that it's meeting that same sort of Moore's law kind of curve that sequencing has done. So finally, people are starting to really think about optimizing this up hugely to how do you make it more accurate, right? How do you have a less error sort of? And we also want to do studies where we might have very limited inputs of samples. So there's a lot of interest in clinical genomes from NICU infants, right? Where you're going to get either a blood spot, like a heel prick amount of DNA. So you really do need sample prep methods that have high conversion efficiencies so that you actually have enough material to load on a sequencer to get a whole genome. So that stuff's happening all the time. And we have, the group is working on this. Okay.
I have one last question, if there's no one in the room who has one, which is that if you were an investigator or advising an investigator who is starting a project where there might be genome sequencing to happen in 5 years, how would you recommend that they stabilize and store those samples, given what you've seen in terms of the technologies advancing and DNA quality standards and changes in preparation?
Sure. And of course, this year, the interest in long reads has gone like crazy. 5 years from now, I imagine it'll be even more interest in long read, even on a population scale, probably at certain clinical genomes. To do that, you can't have super fragmented damaged DNA. So whatever methods are believed to hold that sort of, you may hold it stable in its original biological material. If it's frozen well, but once extracted, you really want that in a state that is not going to fragment over time, and you're not going to degrade your sample over the duration of a 5 or 10 year project for sure.
Makes sense. Thank you.
Yeah.
We have another question online from Aldo R. They say, fantastic presentation. Where can I find these data on the PacBio long read high confidence calls?
I'm sure we have it available. You could reach out to Genomics at Broad. All these analyses that we do, we provide and are open about sharing. So you can reach out to me or Genomics at Broad, and we'll make sure you can get it.
All right. With that, we will thank Brendan one more time.
Thank you, everyone.